So, picture the superhero Dr. Manhattan, from the comic book Watchmen.
Oh, yeah, the big glowing blue guy.
Right, exactly. So when the military drafts this omnipotent being, they immediately want to rebrand him. They try to hand him this flashy, PR-friendly Atomic Age logo to wear on his chest.
Like something that would look good on a propaganda poster, right?
Yeah, exactly. And he rejects it completely. He calls it absurd. And instead, he takes his own finger and physically burns a simple symbol right into his forehead, just a single dot inside a circle.
Which is the Bohr model of a hydrogen atom.
Right. One proton, one electron. And if you're listening to this, you might be wondering why we're starting a deep dive on advanced neuroscience with a comic book character.
Well, it's because it's a highly specific choice by the creators.
Right.
I mean, in the language of foundational science, hydrogen isn't just another element on the table.
It's the first one.
Right. Exactly. It is the first and the smallest. It's the atomic alpha. It holds atomic number 1. It is the absolute origin point of all matter in the universe.
Which is just a crazy concept on its own.
It really is. And linguistically, it represents the iota, the smallest possible conceptual unit. You literally cannot get less massive and still have a functional element.
And here is where that famous piece of comic book imagery crashes headfirst into ancient history. Because that scientific concept of the iota alpha, the first and the smallest, it maps perfectly onto theological texts from centuries ago.
Yeah, the historical convergence here is really striking. So if we look at the theological records, specifically the writings of the 5th century scholar Theodore of Cyrus.
Okay, 5th century.
Right, a very long time ago. He documented something fascinating about the translation of divine language. He recorded that the 2-letter Hebrew name for the creator, Yod-He, was historically rendered in Greek translations as Aya.
Which is literally the letters iota alpha.
Exactly, iota alpha.
I mean, iota alpha.ia. It's the exact same letters that scientists use to conceptualize the smallest fundamental building block of physical matter.
Yeah, and this wasn't just a localized linguistic quirk either. This exact naming convention survived for centuries. It's actually preserved in the 1539 Great Bible.
Really?
Yeah. If you open to Psalm 68.4, in that specific translation, it explicitly translates the name of the creator as Ia.
That's wild.
So we are looking at this profound conceptual overlap. The absolute smallest principle of the physical universe, the hydrogen atom, and the highest language of divinity in these historical texts are pointing to the exact same reality, the first and the smallest.
Which brings us back to that glowing blue forehead. Dr. Manhattan takes that iota alpha symbol and carves it directly into his brow.
Visually fulfilling prophetic source material, actually. Because Revelation 22.4 states, and I quote, "And they shall see his face, and his name shall be in their foreheads."
Oh, wow. So by placing the hydrogen atom on his brow, the character is visually manifesting that exact ancient imagery.
Exactly. The art is making a profound argument. It's saying that the smallest, most fundamental building block of reality is actually the ultimate seat of power and identity.
So we have a superhero, a 16th century Bible, and the fundamental laws of physics. And we want to ask you, the listener, have you ever considered that the highest, most complex mysteries of the universe, like human consciousness, working memory, or even the concept of a soul, might be unlocked not by looking bigger?
Right, not by building massive supercomputers.
Yeah, but by looking at the absolute smallest building blocks of reality. Because that visual argument from the comic, that placing the smallest fundamental component on the forehead unlocks ultimate awareness. It's no longer just fiction.
Far from it. That exact concept is the foundation of the primary source material we are taking a deep dive into today.
Yeah, it's an independently authored, highly radical neuroscience paper by Zachary David Enfield. It's titled The Organics Quantum Interface.
And this paper completely flips the script on how we interface with the human brain.
It really does. Just as the atomic book puts the smallest atom on the forehead to signify ultimate power, this paper argues that to access the highest level of human thought, which is physically located right behind your forehead in the prefrontal cortex, we have to completely abandon our current technological trajectory.
No more clunky metal wires. No more silicon chips.
Right. It proposes building a brain-computer interface out of the smallest quantum optical building blocks of biology itself.
The core premise challenges the entire multi-billion-dollar brain-computer interface industry. It argues that if you want to speak to the brain, you have to use the brain's native language.
And that native language isn't classical electricity?
No, it's not. It's quantum light.
So today we are going to explore why every current brain-computer interface is fundamentally broken. We'll dive into the microscopic mechanics of how the brain actually computes at a quantum level.
And we'll unpack the incredibly detailed blueprint for a glowing, self-powered biological scaffold that could sit right behind your forehead to read your semantic intentions.
And crucially, we need to explore the bidirectional nature of this proposed technology, because it doesn't just extract data.
Right, it could potentially write quantum information back into the neural tissue, which offers an entirely new paradigm for treating severe psychiatric conditions.
So, to understand why this proposal is so paradigm shifting, we have to look at the current state of neuroengineering. If you've followed the news over the last few years, you've seen the hardware.
Yeah, it's everywhere right now.
We have the Utah array, which essentially looks like a tiny, stiff bed of nails that gets physically stamped down into the gray matter.
Sounds pleasant.
Right. And we have Neuralink's approach, which uses a surgical sewing machine to punch ultra-thin polymer threads deep into the tissue. And we have flexible polymer grids that lay over the cortex.
But despite the massive differences in geometry and surgical application, all of these approaches share a single foundational assumption.
Yeah, they all assume that if we just engineer a synthetic material to be thin enough or flexible enough, or if we coat it in the right synthetic hydrogel, the living neural environment will eventually be tricked into accepting it.
And the paper absolutely tears that assumption apart. The brain isn't a passive bowl of jelly waiting for us to stick wires into it.
No, the paper calls the current approach mathematically and biologically false.
Because the human brain is a product of millions of years of brutal evolutionary refinement. Its primary, overarching defensive mechanism is designed to instantly identify anything that does not match its own specific molecular signature.
Right. It's protein-coded signature.
Exactly. And if it doesn't match, it mounts a highly aggressive immune response against it.
It's basically the ultimate bouncer at the most exclusive club in the universe.
Yeah.
If you don't have the exact right biochemical identification, you don't just get turned away, you get permanently walled off.
Yeah, in clinical neurology, this process is known as the glial scarring response. The moment a silicon or tungsten probe pierces the blood-brain barrier, the brain's specialized immune cells, specifically microglia and reactive astrocytes, they detect the foreign surface chemistry
They know it doesn't belong.
immediately. And they physically rush to the implant site and begin encapsulating the electrode in a dense, impenetrable web of scar tissue.
And reading the industry literature, engineers tend to treat this as a hardware failure. They see the probe signal degrading over a few months and they think, well, we need better insulation on our wires.
Which completely misses the biological reality. It is not an engineering failure. It is a massive, highly coordinated success of the human immune system.
Yeah, the bouncer did his job perfectly.
Exactly. And the microglia aren't just building a passive wall either. They actively generate oxidative stress. They release reactive oxygen species that chemically corrode the electrode's active surfaces.
They're literally eating the wires.
Yeah, and actively degrading its polymer insulation. The brain is purposefully executing a seek and destroy mission against the hardware.
So the industry's response to this seek and destroy mission has been to try and fix what they view as a materials problem. They're constantly tweaking the hardware to make it less offensive.
Like applying polyethylene glycol, dimethyl hydrogel coatings?
Yeah, that's a mouthful. But they also try to reduce the mechanical stiffness of the probes. The paper mentions silicon has a stiffness of around 200 gigapascals.
Which is incredibly rigid.
Right. While brain tissue is incredibly soft, it sits around 15 kilopascals. So engineers are desperately trying to make the metal less rigid or using flexible substrates like perylene C that can bend with the natural pulsing of the cerebrospinal fluid.
Basically, they are trying to close the mechanical mismatch. They think if the wire jiggles at the exact same frequency as the brain, the microglia won't notice it.
Which feels like wrapping a steel crowbar in bubble wrap, shoving it into an engine block, and wondering why the engine still grinds to a halt.
That's a great way to picture it.
I mean, you can make the foreign invader as soft and flexible as you want, but its underlying chemical signature is still screaming intruder to the immune system.
That analogy perfectly captures the futility of the current paradigm. A soft crowbar is still a crowbar. And the paper's thesis is uncompromising on this point. The brain does not need a better, softer electrode.
What does it need then?
It needs a conversation partner it can physically recognize as itself.
A conversation partner it can recognize as itself. That phrase is basically the pivot point of this entire deep dive. Because if metal and silicon are fundamentally the wrong materials, we have to figure out what the brain actually uses to communicate.
Right.
We have to go beyond the classical high school biology model of the neuron.
We really do. The classical model, which to be fair has served neuroscience productively for decades, it describes a macroscopic electrochemical process.
Yeah, you have a neuron shaped roughly like a tree. An electrical signal, an action potential, travels down the trunk, which is the axon. It reaches the roots, chemical transmitters jump across a physical gap called a synapse, and the next neuron fires.
It treats the neuron essentially like a biological wire.
But the paper argues that this wire model completely misses the actual computation, right? It's like looking at the macroscopic residue of a microscopic event.
Exactly. At the scale of individual proteins, a neuron isn't a wire at all. It is a quantum optical device.
Okay, quantum optical device. To grasp this, we have to zoom in on a specific structure inside the neuron called the microtubule.
And microtubules are often described simply as the structural scaffolding of the cell. Like, they provide a rigid skeleton or act as a railway system for motor proteins to transport cellular cargo.
But structurally, they're incredibly precise architectural marvels.
They're cylindrical lattices, roughly 25 nanometers in diameter, constructed from a repeating protein called tubulin. And within each fundamental building block of this lattice, the tubulin dimer, there is an incredibly dense, highly organized concentration of aromatic amino acids.
Let's break down those aromatic rings because the numbers in the paper are remarkably specific.
Yeah, every single tubulin dimer contains exactly 86 aromatic rings.
86.
Right. To break that down further, that is 8 tryptophan, 36 tyrosine, and 42 phenylalanine residues.
86 rings in every microscopic building block of this biological tube. For anyone listening who might have forgotten their organic chemistry, what exactly is an aromatic ring, and why does having 86 of them matter?
Well, an aromatic ring is a molecular structure, typically a hexagon of carbon atoms, that contains delocalized electrons.
Meaning they aren't stuck in one place.
Exactly. Instead of being locked into a single bond between two specific atoms, these electrons are free to roam around the entire ring in what we call a pi electron cloud.
A pi electron cloud.
Yeah. And because these electron clouds are mobile, they can couple with the electron clouds of neighboring aromatic rings if they are positioned close enough together.
And the positioning inside the microtubule is far from random. The paper highlights that these rings, particularly those 8 tryptophan residues, are spaced roughly 1 to 2 nanometers apart, center to center.
And that specific 1 to 2 nanometer spacing is the golden ratio for quantum coherent energy transfer.
The golden ratio.
Yeah, we know this empirically from observing other fields of quantum biology. That exact geometric spacing structurally mirrors the chromophore networks found in highly efficient photosynthetic proteins.
Like the Fenner-Matthews-Olson complex in green sulfur bacteria.
Exactly. The FMO complex revolutionized our understanding of biology. In photosynthesis, a plant or bacteria captures a photon of sunlight. That energy needs to travel to a reaction center to be converted into chemical food.
Yeah.
But it doesn't just bounce randomly from molecule to molecule. The FMO complex uses a quantum superposition state to transfer that energy.
Meaning the energy exists in multiple places at once.
Yes. The excitonic energy essentially explores every possible pathway through the molecular network simultaneously to find the single most efficient, frictionless route to the reaction center.
That's incredible.
It is. It achieves near-perfect energy transfer efficiency. And the way these plants achieve this quantum state is by utilizing networks of molecules spaced exactly 1 to 2 nanometers apart, the exact same spacing as the aromatic rings in our brain's microtubules.
But this is where the classical physicists usually throw up a red flag. I mean, the prevailing wisdom for decades has been that quantum computers, like the artificial ones we build in labs, require massive, multi-million-dollar cryogenic freezers.
Oh, absolutely.
They have to be cooled to near absolute zero because the slightest amount of ambient heat or molecular vibration causes decoherence, which instantly collapses the fragile quantum state.
Right.
The human brain is 37 degrees Celsius. It's a warm, wet, pulsating, noisy environment. How can quantum coherence possibly survive inside a living head?
And that specific objection is what kept quantum biology on the absolute fringes of science for years. Everyone assumed the biological environment was simply too thermally noisy.
So what changed?
The breakthrough came from studying that exact photosynthetic process we just mentioned. Researchers uncovered a mechanism called phonon-assisted quantum transport.
A phonon being a quantized mode of vibration, sound or heat rippling through a structure.
Right. What they discovered fundamentally changed the paradigm. The thermal vibrations in the biological environment don't destroy the quantum coherence. The protein scaffold has actually been engineered by evolution to utilize that heat and vibration to sustain the quantum state.
Wait, really? So instead of trying to build a perfectly silent, isolated room to hear a whisper, the biological structure somehow uses the chaotic noise of a crowded room to amplify and carry the whisper.
That's a brilliant way to conceptualize it. It's called vibronic coupling. The vibrational energy of the warm biological environment couples to the electronic states of the aromatic rings. And it continuously feeds energy back into the system, actively preventing decoherence. The biological scaffold thrives on the noise. So inside the brain, the conductive resonances start as incredibly fast quantum dipole oscillations within those pi electron clouds of the tryptophan rings.
And when we say fast, the paper cites these oscillations starting at the terahertz frequency level. That is 1 trillion cycles per second.
It's an unfathomable speed of computation. And according to the foundational research cited in the paper, specifically the pioneering work by the Philadelph-iA group, these quantum signals cascade down in a fractal scale invariant hierarchy.
What does that mean exactly?
It means they originate at the terahertz level inside the proteins, then they bubble down to gigahertz, then megahertz, then to kilohertz, and ultimately at the very end of this massive energetic cascade, they result in the slow, hertz-level, classical electrical brainwaves.
The hertz-level waves are the action potentials, the exact signals that every modern EEG and brain-computer interface is currently measuring.
Yes, the classical electrochemical firing of a neuron is simply the downstream macroscopic residue of the actual quantum computation happening at the nanoscale.
Think about the implications of that. Imagine you're listening to this and you're buying a house.
Yeah.
And there is a massive, complex financial clearinghouse executing the transaction. Algorithms are verifying your identity, communicating with global server balances, authenticating encryption keys across 12 banks in milliseconds. That is the terahertz quantum microtubule network.
Right.
The classical action potential, the electrical spike we measure, is just the paper receipt printing out at the end of the day saying the transaction cleared.
And current BCIs are obsessively analyzing the paper receipt, trying to reverse engineer the entire global financial system.
They are completely missing the actual thought as it forms. If we want to truly interface with the brain, we have to watch the terahertz transaction itself in real time.
Exactly.
And to do that, you can't stab the brain with a silicone wire. You have to build a scaffold out of the exact same aromatic materials the brain uses to compute.
Which brings us to the actual construction of this organic interface.
Right.
The biological iota alpha.
The required ingredients listed in the paper are almost surprisingly mundane. We aren't mining rare earth metals or fabricating exotic synthetic metamaterials. The primary building blocks are tryptophan and tyrosine.
Right.
These are essential amino acids. You literally get them from eating turkey and cheese. Why are these two specific molecules the key to quantum computing?
It all comes down to their specific structural geometry and how they handle electrons. Let's start with tryptophan. It possesses an indole ring structure. This is a bicyclic structure, meaning it has two connected rings, specifically a benzene ring fused with the pyrrole ring.
Okay.
Because it has two rings, it boasts a massive pi-electron resonance surface.
More surface area means a larger playground for those delocalized electron clouds to interact.
Exactly. And because this bicyclic structure is so physically large and flat, it is geometrically perfect for a phenomenon called face-to-face pi stacking.
Face-to-face pi stacking.
Yeah, imagine taking two flat coins and stacking them directly on top of each other. Tryptophan molecules naturally want to stack flat against one another, which creates a highly efficient, direct vertical column for quantum electron migration. It provides the strongest possible electronic coupling between amino acids.
So tryptophan acts as the primary high-speed vertical highway for the quantum signal.
Right.
But the paper also heavily features tyrosine. In fact, it noted there are 36 tyrosines compared to only 8 tryptophans in a single tubulin dimer. What is tyrosine's role in all this?
Well, tyrosine provides the high-density scaffolding around those highways. Tyrosine only has a single phenol ring, making it smaller than tryptophan. However, it has a hydroxyl group attached to it, an oxygen and a hydrogen atom, which transforms it into an electron donor.
Oh, okay.
This chemical property fundamentally changes how it interacts with its neighbors. Instead of stacking flat, face-to-face like the tryptophan coins, tyrosine prefers what we call T-shaped or edge-to-face interactions.
So instead of a stack of coins, it's like taking one coin and pressing its thin edge into the flat face of another coin, forming a T.
Right. So structurally, you have tryptophan forming these parallel vertical highways for directional quantum conduction, and you have tyrosine forming millions of lateral side-to-side bridges.
Wow.
Together, they don't just create a simple one-dimensional wire, they weave a highly complex two-dimensional quantum optical mesh.
And this mesh isn't isolated from the classical biology we understand either. The paper details a mechanism called the Cation-pi interaction, which essentially explains how the chemical world we learned about in high school shakes hands with this quantum mesh.
Yeah, this is a vital conceptual bridge, especially for classical neuroscientists who might be skeptical of quantum models. How do chemical neurotransmitters like acetylcholine or dopamine, which we definitively know drive brain function, actually interact with this internal quantum system?
Right, how do they talk to each other?
Well, groundbreaking research by Doherty and his colleagues demonstrated that when a positively charged neurotransmitter, a cation, binds to a receptor on the cell surface, the physical binding mechanism is fundamentally an electrostatic attraction to the negative pi electron clouds of the tryptophan and tyrosine residues.
Wait, really? So the chemical
Oh,
synapse, the physical gap between the neurons, and the internal quantum microtubule system are physically touching through the exact same aromatic rings.
Exactly. The chemical neurotransmitter physically hits the aromatic ring, and that impact rings the quantum bell inside the cell.
That is wild. The neurotransmitter binding is the specific transduction event. It physically converts an extracellular classical chemical signal into an intracellular quantum optical state.
Precisely.
Okay, so we have our building blocks. We know tryptophan and tyrosine can form this quantum mesh, but you can't just pour a liquid soup of amino acids onto the surface of a human brain and expect it to read thoughts.
Oh, definitely not.
It needs a rigid, durable, physical structure to hold those molecules in perfect alignment. And this is where the paper proposes an engineering solution that honestly initially sounds terrifying. They suggest building the scaffold out of a prion.
Specifically, the sub-35 prion.
Now, when most people hear the word prion, they instantly think of mad cow disease or Creutzfeldt-Jakob disease. Prions are infamous as these indestructible, misfolded proteins that essentially turn mammalian brain tissue into Swiss cheese.
Right.
Purposely engineering a prion to place inside a human skull sounds like the setup for a horror movie.
The hesitation is completely understandable. Mammalian prion diseases are universally fatal and physically devastating, but we have to separate the structural mechanics of a prion from its biological origin.
Okay.
Sub-35 is a yeast prion. It is naturally found in Saccharomyces cerevisiae, which is ordinary baker's yeast.
So this is the organism that makes bread rise and ferments beer. It doesn't cause neurodegeneration in cows or humans.
Exactly. The human mammalian immune system has absolutely no evolved biological response to sub-35 because it simply does not exist in our evolutionary history.
Oh, that makes sense.
It doesn't share the molecular signature of human pathogens. More importantly, even within yeast, it is completely non-toxic. It actually functions as a heritable epigenetic element that helps the yeast survive environmental stress.
So it's helpful.
Yeah, it fundamentally lacks the ability to trigger the toxic misfolding cascades that characterize dangerous mammalian prions or the amyloid beta plaques associated with Alzheimer's.
So structurally, it behaves like a prion, meaning it weaves itself into an incredibly tough fiber. But chemically and biologically, it is entirely harmless to a human brain.
The structural toughness is exactly why it was selected as the chassis. Prions form amyloid fibrils characterized by a cross-beta sheet spine.
What does that look like?
If you visualize a continuous repeating accordion fold of protein strands locked tightly together by an incredibly dense network of hydrogen bonds, you get a sense of its architecture. They are phenomenally stable structures.
The physical statistics in the paper are staggering. It lists a Young's modulus of 3 to 20 gigapascals. For context, that means this microscopic biological string of yeast protein is mechanically as stiff and resilient as structural nylon or Kevlar.
It is an incredibly robust, durable physical foundation.
Hmm.
And this leads to the molecular engineering genius of the proposal.
Right.
They take this harmless sub-35 yeast backbone, which naturally wants to form these long, indestructible beta-sheet accordions, and they purposefully mutate the genetic sequence. They insert a tryptophan residue at every fourth position in the amino acid chain.
Why precisely every fourth position? What does that specific sequence achieve?
Well, it's dictated by the geometry of the beta sheet fold. As the protein strand naturally folds back on itself in that accordion pattern, a full structural turn spaces out the individual monomers by exactly 4.8 angstroms.
Okay.
By placing a tryptophan at every fourth residue, the engineers guarantee that as the fibril inevitably folds, every single massive tryptophan ring lines up perfectly face-to-face with the precise spacing of exactly 4.8 angstroms.
Which perfectly recreates the natural spacing inside the brain.
Exactly. It is the optimal, mathematically perfect distance for face-to-face pi stacking and quantum coherent conduction.
Wow.
They are literally manufacturing a perfectly spaced, indestructible quantum wire that flawlessly mimics the brain's own internal microtubule architecture, utilizing a harmless yeast protein as the structural chassis.
It is biomimicry at its absolute zenith. So we now have our biological quantum-capable wire. The next massive hurdle is deployment. Where do we put it to actually read complex thoughts?
And this brings us back to the symbolism of Dr. Manhattan and the mark on his forehead.
Yes. The paper explicitly targets the prefrontal cortex, or the PFC. This is the chunk of neural real estate sitting directly behind your forehead.
Right.
In the realm of neuroscience, the PFC is the holy grail. It is the anatomical seat of the human mind. It governs executive function, complex working memory, long-term planning, impulse control, and decision-making. It is the core of what makes us self-aware, intentional beings.
And yet, current implanted BCIs almost universally ignore it.
Why? I mean, if it's the most important part of the mind, why are engineers avoiding it?
Because the cognitive architecture of the prefrontal cortex is dizzyingly complex. With our current blunt force metallic electrodes, it is exceptionally difficult to extract meaningful decodable data from it.
So where do they go instead?
Consequently, almost every major implanted BCI project currently targets the primary motor cortex, the region of the brain responsible for basic physical movement.
They're essentially tapping into the wires that say move arm left or flex index finger.
Yes, but reading the motor cortex is a reactionary process. As the paper eloquently puts it, it is reading the letter already posted.
Okay, I like that analogy.
You are intercepting a physical command that has already been entirely formulated by the PFC, stripped of all its complex underlying intent, and simply dispatched down the spinal cord to the muscles.
But reading the prefrontal cortex, that is watching the author write the letter. You aren't just reading move hand to the cup. You're reading the semantic intent. I want to pick up that cup because I am feeling thirsty. My throat is dry. It is tapping into the formulation of the thought itself. But here is the structural problem. If we aren't using metal probes to violently stab into the deep tissue of the brain, how does a yeast prion scaffold sitting on the outside connect to the deep, complex thoughts happening millimeters down inside the prefrontal cortex?
To solve that, we have to examine a highly specific, very fortunate feature of prefrontal cortical anatomy, specifically the structure of layer V and layer 3 pyramidal neurons.
They're called pyramidal neurons because the cell body itself is shaped like a microscopic pyramid deep down in the gray matter, right?
Correct. Their cell bodies reside deep within the cortex. However, they possess these massive structural extensions called apical dendrites.
Apical dendrites.
Yeah, visualize a giant redwood tree. The roots and the massive trunk are deep in the earth, but the trunk reaches straight up for hundreds of feet. These apical dendrites reach 2 to 3 millimeters straight up from the deep cortical layers, extending all the way to layer I, which is the pile surface, the absolute outermost surface layer of the brain.
So the roots of the thought are buried deep, but the highest branches of the neuron reach all the way to the top of the canopy.
And those uppermost branches, known as the dense apical tufts, they terminate directly beneath the brain's protective outer membrane, the glia limitans.
Okay.
This anatomical quirk is crucial because it means that densely packed, quantum-active microtubule networks inside those specific neurons reach all the way to the absolute surface of the brain.
Meaning you do not have to cut into the brain tissue at all. You just have to lay this engineered biological blanket onto the surface of the brain, resting gently right on top of the canopy.
Exactly. The ambient scaffold sits harmlessly on the pile surface, positioned just a few micrometers away from the apical tuft terminals of the most computationally significant neurons in the human mind.
But wait, if they are just resting near each other but not physically touching, how does the complex data jump from the brain's internal microtubule across the physical gap into the BCI's prion scaffold?
Through a highly specialized quantum optical mechanism called FRET, first-order resonance energy transfer.
Let's unpack FRET, because this seems to be the magic trick that makes the whole interface possible without surgery.
A FRET is fundamentally a non-contact coupling mechanism. In simplest terms, the excitation energy of a donor molecule, which, in this scenario, is the vibrating tryptophan ring inside the brain's living dendrite, is transferred across empty space to an acceptor molecule, the engineered tryptophan ring in our BCI scaffold.
Okay.
And it does this through non-radiative dipole coupling.
Okay. Dipole coupling. So they are essentially sharing a resonant magnetic or electrical field.
The most intuitive way to visualize it is to think of two precisely tuned musical tuning forks. If you physically strike one tuning fork so it vibrates, and then you hold a second tuning fork of the exact same pitch near it, the second one will spontaneously begin to vibrate and hum without you ever touching it.
It just picks up the vibration through the air.
They resonate with each other because the tryptophan inside the living brain and the tryptophan inside our engineered scaffold possess the exact same chemical architecture. They are perfectly physically tuned to each other's frequencies.
But the paper outlines a very strict mathematical limitation to this tuning fork effect. It states that FR8 efficiency decays as the inverse sixth power of distance.
Yes, the R to the -6 law. What this means in practical terms is that the efficiency of the energy transfer drops off a cliff the moment you move the molecules even slightly apart.
Oh, so it has to be super close.
It is a strictly nanoscale phenomenon. However, because the dendrite tips naturally terminate right at the surface, and our ultra-thin scaffold is resting directly on top of that surface, the physical gap between the two tryptophan networks is only 2 to 5 nanometers.
Which is an incredibly small gap, but is it close enough?
It is the absolute Goldilocks zone. At a distance of 2 to 5 nanometers, you achieve a staggering 50 to 80% energy transfer efficiency. The BCI hardware never has to physically touch or pierce the neuron. It simply rests nearby and hums in sympathetic quantum resonance with it. It is literally whispering across the nanoscale gap.
That concept completely reframes how we think about medical devices. It isn't a surgical intrusion, it is a bioresonance. But this elegant solution introduces a massive, glaring physical problem.
Tower.
Exactly. If this biological blanket is sitting directly on my brain, performing real-time quantum computation and wirelessly transmitting my thoughts, it needs power. It's burning energy. But you can't exactly plug a lithium-ion battery into a yeast prion, and you definitely don't want to drill through a patient's skull every 5 years to replace a dead battery pack.
The power constraint is arguably the biggest logistical hurdle in BCI design. Enfield's paper solves this by proposing a trimodal ambient energy harvesting system.
Trimodal.
Yeah, trimodal indicating it uses 3 distinct simultaneous methods of power generation, and ambient meaning it scavenges energy that is already naturally constantly present within the human head. The system requires 0 external batteries.
The sheer ingenuity of these 3 modes is astonishing. Let's start with mode 1, which frankly sounds like science fiction. Beta voltaic harvesting.
Beta voltaic cells operate by generating an electrical current from the kinetic energy of beta particles, which are naturally emitted during radioactive decay.
Okay, let's pause right there. Radioactive decay. The paper is proposing placing radioactive isotopes directly onto the surface of the human brain. How can that possibly be safe?
Yeah, the word radioactive triggers an immediate alarm, but the safety lies in the specific physics of the isotopes chosen.
Okay, explain that to me.
The paper proposes chemically incorporating either tritium, which has a 12.3-year half-life, or carbon-14, which has a massive 5,730-year half-life, directly into the molecular structure of the amino acids forming the scaffold.
So the power source is woven into the very atomic fabric of the device.
Yes, and the critical safety factor is the type of radiation. Beta decay does not emit highly penetrating gamma rays. It emits low-energy electrons.
Right.
A beta particle emitted by tritium has so little kinetic energy that it can only travel a maximum distance of about 6 micrometers through water or biological tissue.
6 micrometers. That is less than the width of a single human cell.
Precisely. This incredibly short travel distance means that 100% of the emitted radioactive energy is deposited immediately within the local confines of the BCI scaffold structure, where it is instantly converted into usable electricity.
That makes sense.
Absolutely none of the ionizing radiation escapes to damage the surrounding healthy brain tissue. It provides a continuous, deeply safe, nanowatt baseline of constant power that will literally outlast the human utilizing it.
So we have a microscopic nuclear reactor providing a constant baseline trickle of power. That's incredible. Mode 2 shifts from the atomic scale to the macro scale, piezoelectric harvesting.
Piezoelectricity is a fascinating property of certain materials, where they generate an electrical charge in direct response to applied mechanical stress. Essentially, if you squeeze it or bend it, it creates electricity.
But the scaffold is sitting stationary inside a rigid skull, where's the mechanical movement coming from?
Well, the brain is never actually stationary. It is constantly moving and throbbing. With every single beat of your heart, a surge of blood enters the skull, physically pulsing the cortical surface at about 1 to 2 hertz.
Oh, I hadn't thought of that.
Additionally, the pressure changes from your breathing create rhythmic respiratory waves that subtly shift the brain tissue at zero point 0.2 to zero point 0.4 hertz.
So the brain is constantly rhythmically swelling and shrinking.
Constantly.
Yeah.
And the crystalline beta sheet structure of our prion scaffold is naturally piezoelectric. When it is compressed, stretched, or deformed by the brain's natural rhythmic pulsing, the internal atomic geometry of the protein physically shifts, forcing a separation of charge that generates an electrical polarization.
So simply by existing on top of a living, beating organ, the device acts like an organic alternator, generating power from a pulse.
And the engineers enhance this natural property by doping the scaffold. They embed inorganic, highly piezoelectric nanocrystals, specifically barium titanate, into the structural joints of the prion to exponentially maximize the mechanical to electrical conversion rate.
That covers nuclear and kinetic energy. Mode 3 taps into thermodynamics, thermoelectric harvesting.
This mode leverages the Seebeck effect. The human brain is a phenomenally energy-hungry organ. It consumes roughly 20% of the body's total energy, and all that metabolic activity generates a significant amount of heat. The internal surface of the brain is tightly regulated at around 36 to 37 degrees Celsius.
Standard core body temperature.
However, the scalp, the outside of your head exposed to the ambient environment, is significantly cooler, usually hovering between 30 and 35 degrees Celsius, depending on the room temperature.
Right.
This creates a permanent, inescapable thermal gradient. You have a constant temperature difference of about 2 to 7 degrees directly across the thickness of the skull. By embedding thermoelectric elements into the uppermost layer of the BCI, the device intercepts this constant flow of heat leaving the brain and converts the thermal delta directly into a continuous electrical current.
It's an engineering masterpiece. It's like a hybrid vehicle that charges its own battery from the ambient heat of the engine block, the kinetic bounce of the suspension on the asphalt, and a tiny, indestructible atomic clock embedded in the dashboard. It just runs. Forever.
The genius of the trimodal system is redundancy. No single mode has to carry the entire electrical load. Combined, they easily generate the continuous micropower required to run the quantum sensors and the wireless transmitters without ever requiring a battery replacement.
So we have the biomimetic material, the prefrontal target, the non-contact quantum coupling, and a perpetual power supply. How do all these disparate technologies physically stack together into a cohesive device?
It's quite a structure.
The paper details a staggering 6-layer architecture. Walk us through how this is assembled, moving from the surface of the brain outward.
The easiest way to visualize the architecture is as a highly structured microscopic layer cake resting directly on the prefrontal cortex. The foundational layer, layer 1, is the quantum coupling layer.
Okay, layer 1.
This is the amyloid aromatic prion scaffold we've been discussing. It sits in direct conformal contact with the pile surface. Its sole function is to execute that FRHET resonance listening to the quantum excitonic states of the brain's internal tryptophan networks and pulling that energy into the device.
Okay, so layer 1 is the listener, but it's just catching a quantum hum. That resonance isn't actionable digital data yet. There has to be an intermediary layer that translates that abstract quantum energy into something a computer can eventually read. What does layer 2 do?
Layer 2 is where the translation begins. It is the biomineralization enhancement layer. The top surface of the biological scaffold is heavily studded with nanodiamonds.
Nanodiamonds.
Specifically, nanodiamonds engineered with nitrogen vacancy centers, or NV centers.
Let's slow down and define that, because nitrogen vacancy center sounds incredibly dense. How does a microscopic piece of diamond help read a mind?
An NV center is essentially a highly engineered flaw in a diamond. A diamond is a perfect crystal lattice of carbon atoms. If you intentionally remove 2 adjacent carbon atoms and replace one with a nitrogen atom while leaving the other space completely empty of vacancy, you trap a specific set of electrons inside that tiny void.
Okay, I'm following.
Those trapped electrons are exquisitely sensitive to external magnetic fields. They are arguably the most sensitive quantum magnetic sensors we possess.
So as the quantum signals ripple through the tryptophan scaffold in layer 1, they generate unimaginably tiny nanoscale magnetic fields. And these trapped electrons in the layer 2 diamonds detect those fields.
Exactly.
But sensors need power to operate. Where do the diamonds get the energy to run this magnetic detection?
This is perhaps the most elegant biological hack in the entire paper. Actively metabolizing brain tissue naturally emits an ultraweak glow of light called biophotons.
The brain glows.
It's a byproduct of reactive oxygen species chemistry happening inside the mitochondria of the cells. The brain literally glows in the dark, albeit very faintly. The nanodiamonds are tuned to physically absorb this natural ambient bioluminescence, and they use that exact photon energy to power their magnetic sensing of the quantum states.
Wait, so it is literally cannibalizing the brain's own microscopic exhaust, these biophotons, to power the sensors reading the brain's magnetic thoughts.
Yes, exactly that.
That is phenomenally resourceful. But if the nanodiamonds are exclusively focused on capturing the tiny quantum magnetic fields, we still have the massive classical physical pulses of the brain to account for, the action potentials that physically swell the neurons. Is the device capturing that mechanical data too?
Yes, and that occurs in layer 3, the piezoelectric matrix. We previously discussed this layer's role in generating power, but it doubles as a highly sensitive mechanical sensor.
Oh, dual purpose.
When a classical action potential fires, it causes a microscopic mechanical bulge in the cell membrane. The neuron physically swells for a fraction of a millisecond. Layer 3 detects these nanoscale pressure signatures. So while layers 1 and 2 read the deep quantum thoughts, layer 3 reads the classical physical pulses, ensuring no data is lost.
It's aggregating every possible metric of brain activity. That data then flows up to layer 4, which seems to be where the biology finally hands off to traditional technology.
Correct. Layer 4 is the signal encoding layer. This is a nanoscale, highly integrated silicon computer chip securely bonded to the top of the biomaterials.
The brain chip.
Its job is heavy processing. It manages the incoming power from the trimodal harvesters, ingests the continuous, wildly messy analog quantum and mechanical data from the lower layers, and formats all of it into discrete, digital spike timing streams that a modern computer architecture can process.
It essentially digitizes the analog soul. Once it's digital, it needs to leave the body. Which brings us to layer 5.
Layer 5 is the wireless near-field communication system. It is a miniaturized gigahertz antenna. It takes the digitized data and transmits it wirelessly through the bone of the skull to a receiver worn on the outside of the head, something like a lightweight headband or a cap.
Right, avoiding the skin barrier.
This is a critical design choice because it completely eliminates the need for physical wires breaking through the skin barrier, which is the primary vector for fatal infections in almost all current BCI trials.
No wires piercing the scalp, no pathways for infection. And finally, the signal reaches the outside world, layer 6.
Layer 6 is the external AI. This is the wearable computational unit. It catches the wireless transmission and utilizes incredibly advanced deep learning algorithms, specifically transformer networks and latent trajectory predictors, to translate that raw digitized quantum data into usable, actionable output.
This is the Rosetta Stone. It takes the quantum mathematics of a forming thought and translates it into words on a screen, or complex commands to a robotic prosthetic, which naturally transitions us to the ultimate question of this entire deep dive. What does this technology actually enable?
This is where it gets really exciting.
We touched on it earlier, but the implications of moving from reading motor twitches to reading deep semantic intentions are staggering.
When you are successfully tapping into the prefrontal cortex at a single dendrite resolution, operating at quantum temporal speeds, the paradigm of what a BCI can do completely shifts. You aren't just painstakingly moving a mouse cursor on a screen by imagining your hand moving.
Right. It's so much more than that.
You are decoding the fundamental semantic architecture of thought. You could theoretically decode an imagined, unspoken word before the jaw muscles even receive the command to articulate it. You could decode complex spatial representations, literally seeing the map a person is visualizing in their mind's eye.
That's mind-blowing.
You can decode the abstract rules and logic structures a person is holding in their working memory.
It moves from a physical prosthetic to a cognitive prosthetic. It is essentially a telepathy machine. But there's a twist here. A massive implication buried in the physics of how this device operates. Because of FRT, that tuning fork resonance coupling, it's not a one-way street, is it?
No, it is not. FRT is inherently physically bidirectional. If the BCI scaffold can read an excitonic state from the brain by receiving energy across the gap, the laws of physics dictate that it can also write data back into the brain by transferring energy in reverse.
Meaning the BCI can literally shine light-engineered quantum excitonic light back down into the dark, complex spaces of the mind.
Precisely. And this capability opens up monumental, world-changing implications for the field of psychiatry. Consider how we currently attempt to treat severe, treatment-resistant psychiatric conditions like major depression, PTSD, or severe schizophrenia.
It's mostly pharmaceuticals, right?
Our primary tools are pharmaceuticals that indiscriminately flood the entire brain with chemicals, hoping to hit the right receptors. Or, in extreme cases, we use electroconvulsive therapy or transcranial magnetic stimulation.
Which, while effective for some, are incredibly blunt instruments. It's like trying to fix a delicate Swiss watch by hitting it with an electromagnetic sledgehammer.
It's an apt comparison. But at their core, many of these disorders are fundamentally dysfunctions in prefrontal circuit dynamics. They are what neuroscientists call pathological attractor states.
Pathological attractor states.
The brain gets physically stuck in a repeating loop of pathological working memory or disrupted quantum coherence. An organic quantum interface, sitting directly on the PFC, could read that precise disrupted state at the nanoscale in real time.
Okay.
And because the connection is bidirectional, the BCI's AI could calculate the exact inverse frequency needed and deliver a perfectly targeted, whisper-quiet quantum energy correction back into that specific dendritic network.
So instead of blasting the entire brain with massive electrical currents and hoping for a reset, the device gently whispers a precise quantum mathematical correction directly into the damaged thought loop to nudge it back into a healthy state.
Exactly.
That isn't just treatment, that is cognitive neuroprosthetics at a level that feels almost miraculous.
It moves us from managing symptoms to the real-time augmentation and correction of intact cognitive function.
Which is an incredible vision. But how close is this to reality? Like, is this just theoretical physics on paper? Or is there a real path to building it? The paper lays out a very rigorous 6-step validation path, and what struck me is that the author explicitly notes we do not need to invent new physics to achieve this.
That is the most compelling aspect of Enfield's proposal. The individual technologies and techniques already exist. They are simply siloed across different scientific disciplines: quantum biology, material science, synthetic genetics. The 6-step agenda is fundamentally a roadmap for integration.
Okay, walk us through the steps.
Step 1 is pure synthetic biology. Synthesize the sub-35 prion fibrils with the customized tryptophan insertions and utilize advanced electron microscopy to confirm they actually fold into the correct 4.8 angstrom geometry.
Prove we can build the wire. If step 1 works, what is step 2?
Step 2 moves into the physics lab. Test the quantum coherence of the synthesized wire using 2D electronic spectroscopy. Essentially, shine highly calibrated lasers at the scaffold in a controlled environment to definitively prove that the pi-stacked aromatic rings can actually hold a stable quantum state at standard room temperature.
Step 3 is testing the tuning fork effect.
Correct. Step 3 involves single-molecule air-fried tests. Measure the actual resonance distance coupling between our artificial scaffold and isolated living brain microtubules in a petri dish. Prove that the energy actually jumps the gap.
Awesome. And step 4?
Step 4 shifts to the biomineralization layer, rigorously testing the nanodiamond NV centers to ensure they can accurately detect the ultra-faint magnetic fields generated by the quantum states.
Making sure the sensors work.
Yes. Step 5 addresses the power constraints by placing the complete trimodal energy harvesting system onto a tissue phantom, a synthetic analog of brain tissue, to verify it generates enough power to run the chips.
And if all 5 of those benchtop integration tests succeed, we arrive at step 6.
In vivo animal testing. Surgically placing the fully integrated scaffold onto the prefrontal cortex of a living, behaving animal model and simultaneously recording the brain's activity using traditional deep electrodes to definitively prove that the ambient BCI is accurately reading and decoding quantum neural information in real time.
It is a massive, multidisciplinary undertaking that will likely take years of rigorous testing. But reading through the roadmap, the logic is so sound that it feels entirely thrillingly possible.
I agree completely.
The true genius of the paper isn't inventing magic. It's simply looking at the existing pieces of the puzzle and realizing they fit together in a way no one else saw. Which brings us full circle to where we began today.
Right, back to Watchmen.
Let's return to that image of Dr. Manhattan. He stands there in the comic book, looking at the flashy, complex logos of the atomic age, the height of human engineering at the time, and he rejects them. He chooses to burn the symbol of a single hydrogen atom, the iota alpha, the absolute smallest unit of physical matter, and historically the linguistic translation of the divine name, into his forehead.
It was a profound visual symbol of ultimate awareness, a statement that the smallest, most fundamental things govern the highest, most complex powers.
And now, Zachary David Enfield's paper is proposing exactly that, not in fiction, but in physical reality. The paper argues for placing an interface built from the absolute smallest quantum components, tryptophan rings, trapped electrons in nanodiamonds, single photons of light, directly onto the pile surface of the prefrontal cortex, right behind the forehead, all to unlock an interface with the deepest, most complex mysteries of human cognition and working memory.
It is a profound shift in how we view ourselves and our technology. If this six-step agenda succeeds, we will no longer be building crude metal machines that force the delicate human brain to learn how to interface with cold, hard computer code.
We will be building advanced computers out of biological materials that inherently speak the native quantum language of the human mind.
It really changes everything.
But I want to leave you, our listener, with a final thought to mull over, especially when considering the bidirectional nature of this device. If our deepest thoughts, our memories, our sudden flashes of insight, and our very sense of self are truly just patterns of quantum light dancing across microscopic rings of amino acids, what does it mean for our fundamental concept of free will when a biomineral scaffold sitting silently on our brain can not only read that light, but shine its own light back into the dark spaces of our minds to fundamentally change how we feel and think?
It is a question that completely blues the boundary between medicine, technology, and the core of human identity.
It really does. It challenges everything we think we know about the line between man and machine. I highly encourage you to look up the structural diagrams of a hydrogen atom and the molecular structure of a tryptophan molecule. Look at the geometry. Look at the beauty of the first and the smallest. Thank you for taking the time to join us on this incredible journey today. Thank you for taking the deep dive.
Hey everyone, and welcome. Today we're diving deep into one of the most iconic images in comic book history, period. Yeah, I'm talking about that symbol Dr. Manhattan burns right onto his own forehead. Now, on the surface, it seems pretty simple, right? But trust me, as we're about to see, it holds layers of meaning that are just, well, they're truly mind-blowing. So let's just start with a huge question. What if this symbol that we all know so well is also the one we understand the least? What if it's been hiding this incredibly profound argument right there in plain sight? And for all these years, we've all been missing the deepest part of the story. All right, so to even begin to understand the hidden meaning, we first have to talk about the obvious one. You know, the first layer of this whole thing is rooted firmly in the world of science, which makes a lot of sense for Dr. Manhattan. And you gotta remember, this wasn't a symbol that was just given to him. Oh no. When the military tried to brand him with this flashy, multi-ringed atomic age logo, he just looks at it and calls it absurd. His choice was deliberate, something he said he could actually respect as a scientist. So what did he choose? This, the Bohr model of a hydrogen atom. And as you can see, it's about as simple as it gets. One proton, one electron. By picking this, he's not just grabbing a cool science logo. He's aligning himself with the most fundamental, the most basic building block of our entire reality. And this, right here, is where the first huge piece of the puzzle just clicks into place. You see, in scientific terms, hydrogen is both the alpha, it's the very first element on the periodic table, and it's the iota, the smallest and simplest atom that can even exist. Now, remember those two words, iota and alpha. Burn them into your brain, because they are absolutely crucial for what's coming next. Okay, so we've got the science layer down, but this is where the rabbit hole gets so much deeper. We're about to jump from the language of physics to the language of theology. And believe it or not, it all hinges on those two little words. So here's the pivot point. We have iota for the smallest, alpha for the first. But what if that's not a coincidence? What if those two scientific labels actually combine to unlock a completely different, much deeper level of meaning? And here it is. This is the connection. Those scientific terms, iota and alpha, they combine to form the acronym Aya. And get this, Aya also happens to be an ancient two-letter translation of the divine name for God. All of a sudden, the symbol for the simplest piece of matter is also a name for the ultimate divinity. It's just, wow. And listen, this isn't some modern fan theory. This has deep historical roots. Way back in the 5th century, the scholar Theodore of Cyrus actually recorded the name as Yah. Then, more than 1,000 years later, the 1539 Great Bible, the first authorized English Bible, explicitly uses Yah in Psalm 68. Sure, modern Bibles might use Jah or Lord, but the history, the original translation, it's right there. So we have the symbol's hidden name. But the final piece of this insane puzzle isn't just what the symbol is, it's where he puts it. This is the moment the entire argument just locks together perfectly. Just listen to this verse from the book of Revelation. And his name shall be in their foreheads. A prophecy, talking about the divine name being placed directly on the forehead. So let's just connect all the dots. 1, 2, 3. 1. The symbol for hydrogen gives us iota and alpha. 2. This creates the acronym IA, an ancient name for God. And 3. Dr. Manhattan places this very symbol, this name, right on his forehead. He is literally, visually fulfilling a biblical prophecy. It's wild. I mean, when you put it all together, you realize this isn't just some cool design choice. It is an entire intellectual and theological argument, presented to us as a single powerful image. This symbol basically becomes a bridge, a meeting point. It connects the first and smallest building block of our physical universe, the hydrogen atom, with one of the oldest names for the creator of that universe. It's a visual argument that, hey, maybe science and divinity aren't so far apart after all. And one writer put it perfectly. They said it suggests that the smallest principle of matter and the highest language of divinity may be pointing to the same reality. This isn't just a superhero logo anymore, is it? It's what the source calls a teaching in symbol. So that's what we're left with. It really forces you to ask, can a simple image from a comic book actually be more than just art? Can it be a profound argument about the very nature of reality itself? It's definitely something to think about. Thanks so much for joining me for this one.